Okie dokie. How about we go ahead and get started? So, good afternoon, everyone. My name is Ray Slager, and I'm from Yahara Software. I'd like to welcome you today to our conversation, the next boom in drug discovery, what's missing, and what comes next. Got a couple of quick notes for you. As you're watching, feel free to submit your questions via the chat and Q&A features for our speakers to address. Today's presentation will be recorded and a copy will be shared with all of you. And at the end, you'll be prompted to fill out a quick survey. Please take a minute to do so to help us continue to produce great educational content like today's presentation. I want to welcome you to our speakers for the day. We've got Josh, James, and Grant joining us. Josh is our Director of Engineering here at Yahara Software. James is our Bioinformatics Practice Manager, and we've got Grant joining them, who is a computational biologist, mycologist, and molecular geneticist. Before we go into our presentation, I've got a couple of poll questions for you that I'm going to pull on up here. So first, we want to know, let's get that one going. Which aspect of natural product drug discovery interests you the most? I'm going to pop the answers up on the screen in 3, 2, 1. Looks like our winner today is technological advances like genomics, AI, and, I'm going to say it wrong, metabolomics?
Metabolomics.
Metabolomics.
Yep.
Metabolomics.
I'm right there with you, Ray.
I'm marketing. I'm no scientist. All right. One other question for you.
I say anybody hanging out with us gets to be an honorary scientist today.
Sweet. Second question, what do you think is the biggest challenge in drug discovery today? All right, counting down in 5, 4, 3, 2, and 1.
Ooh, pretty split.
Pretty spread out, yeah. Discovering known compounds, lack of data for AI-driven discovery, and ethical and regulatory concerns. That's it from me. I'm going to go ahead and pass the mic on over to you, fellas.
Awesome. Thank you so much, Ray. And Grant, welcome. Thank you so much for joining us today. And to everybody in the audience, we are glad to have you here. We are going to spend just a little bit of time. Grant's going to give us an introduction to some natural product drug discovery ideas, and then we're going to open this up and have a little bit of a conversation. The reason we've got Josh in is to talk some of those challenges about, okay, lack of data, and I want to apply a machine learning model or an AI model to it. What are some of the opportunities that are here? What are some of the things we can think about? But I think let's start with a little bit of an intro from Grant. Grant, welcome. Thank you for joining us.
I'll introduce myself, and then I kind of wanted to do like a primer on what we're talking about. My name is Grant Nichols. I'm a 5th-year PhD student at UW-Madison, where I've been doing kind of drug discovery, natural product research for the last 5 years. But before joining grad school, I've worked for a couple companies and done some like biofuel research. Natural products. So if you think of like drug discovery as this overarching goal, like we're trying to find new compounds, there are different strategies to finding new drugs. And natural products is just one of those strategies, but it's not the only strategy. And natural products is just this idea that all organisms produce compounds, right? Plants, bacteria, fungi, and specifically for plants, bacteria, and fungi, the compounds that they make are really how they interact with the world, right? They don't have hands, they don't have feet. And so if they need to fight off competitors or if they need to communicate to different organisms, they do it by making compounds. And humans have basically realized that, wow, these organisms are way better at making these cool compounds than we are at just like coming up with them from scratch. And we've realized that we can basically extract these metabolites and find potential drug candidates. So what I'm showing here are specifically like fungal ones, because my lab focuses on fungal natural products, but of course there's plants, bacteria, snake venom, natural products even. But if you look at all of the drugs that are on the market, right, by the FDA, over 50% are directly or indirectly derived from some sort of natural product source. So natural products as a way of like finding new medicines is by far the biggest category. It's the biggest way that we've found success. And specifically 18% come from fungi. I just had to point that out because I'm a big fungal person. I'm just showing here on the slide some of the three like biggest ones. Penicillin probably being the most famous, the one that you guys have all tried. And it comes from the organism Penicillium. So the compound was named after the organism. One other primer before we go deeper into like AI and other things is I want to try to have us not anthropomorphize these organisms. So I want you to all imagine you're a fungus, right? If someone takes you out of the environment and they put you on this nice, cushy, plate in a lab setting where you have all the nutrients you need, the temperature is perfect, there will be no need for you to produce the vast majority of the compounds that you can make. I can almost like, the analogy I would make is like, think of yourself, like if your body is in a AC house, it would be bad if you just started sweating all the time because you only want to sweat when you're hot. Like there's certain things that you want to only happen when you have environmental triggers. And it's the same thing for these organisms. And so a lot of these organisms, what we've realized, will only make about 10 to 20% of the compounds that they can make in a lab setting. And we can dive deeper into like strategies that the community has come to try to get around that. But that's like a big hurdle in natural product drug discovery. Most of the drugs that organisms can make will just never be turned on in a lab setting. And so I feel like that's a good enough of a primer. We can like dive deeper into specific aspects or questions.
I think that sets the stage for us. Where I think I want to go first is when we were preparing for this, you were talking a little bit about the history of how we got here and what natural product drug discovery has looked like historically. Take us through just, what's the 1 minute version of what did that used to look like? And then we'll transition. We'll talk a little bit more about what does it look like.
Sure. Ray, could you go to slide 8? This was one of my hidden slides, but there's a diagram on it that might be kind of helpful. In the top right corner, oh, sorry, go back one. This one. So yeah, on the top right, this is what I'm going to be referring to. Essentially, in my like grandparents' generation, that is what was called like the golden age of medicine discovery and the golden age of natural product discovery. They were finding like compounds every week in that generation. And that's because they were starting to realize we can just grab these things from the wild, extract their metabolites, and find new drugs. And that's pretty much how most drug discovery in the early stages was driven. It was like a chemistry driven thing where you extract out the metabolites and you look for a given bioactivity that you're wanting. But what started to happen is we started rediscovering the same compounds more and more as time went on, where like all of the low hanging fruit kind of got picked by my grandparents' generation. And now it's really difficult to find new drugs today. And the diagram that I'm showing on the top right is showing the rate of new antibiotic discovery. So it's just like one example, but I think it exemplifies like the boom and bust that I'm talking about over time. And you can see it peaked quite heavily in like 1940s to 1950s and then started to die off until today. We have not been able to, as like a society, find a new antibiotic class in the last 20 to 30 years. And most of that comes from the fact that we're just rediscovering the same compounds. Then, I can go into the genoma because there's now another boom, but yeah.
Well, go for it.
Yeah. So, I mean, that was kind of like historically, right? It was driven by the chemistry approaches. And then between like 1980 and 2000, genomics and DNA sequencing started becoming a thing. And specifically in the early 2000s, when people started realizing how to do like full genome sequencing, I think the first genome was sequenced in 2003 from the Human Genome Project. We started looking within those genomes and realizing, wow, there are way more drug pathways within these organisms than we're seeing from a chemistry standpoint in a lab. And that's when we started to realize, wow, these organisms are really only making 10 to 20% of the compounds that they can make in a lab setting. And we know that because we can look at the pathways within the genome itself. But just seeing the pathway doesn't mean it's going to be turned on, right? And so, yeah, for the last 20 years, pretty much I would say genomics and big data in the genomics world has been driving like the natural products field and academics within the field.
And I think that's sort of the first theme here is we needed the advancement to get a lot more data underpinning these kinds of things in order to start getting that next series of advancements.
Definitely.
And I know where you're sort of going. Let's go back to your second slide. The thing to start to talk about here is, okay, I've got genomics, I've solved all the problems. This will give me all the answers. Talk to me a little bit about why that's not necessarily true.
Sure. Ray, can you go one slide in the future? This one. Okay. Um, so yeah, this is we're gonna do some like intro biology. If you guys do not know what the central dogma is, that's fine. I'm gonna try to explain it, or this might be a refresher for some of you guys' like freshman high school classes. Um, this is kind of how cells work. You go from DNA. DNA is transcribed into RNA, which is translated into proteins, which turns into metabolites, which then does things. But like I'm going to simplify it with an analogy. So the DNA you can think of as like a cookbook, right? It has all of the instructions. And so anything pertaining to DNA is what we refer to when we say genomic. Different omics really just mean like different cellular data types. The RNA would be like the individual recipe within the cookbook, right? It's not the full thing, but it's just like the one instruction manual. The protein is like the actual food. Okay, you've made the recipe, you've done the thing, and this is what you're eating. So it's like the actual product. But in natural product discovery, you have to go a little bit further. For most people, a central dogma, they stop at like DNA to protein. Really, proteins make things like in a cell. And so in our case, those proteins are making the drug candidates that we're actually interested in. So we really don't care about the proteins. We want the metabolites. And then even beyond that, it's one thing to like know what a metabolite is. It's another thing to know what that metabolite is doing in a human body, right? Like, is it actually going to be useful as a drug target? And every step of this like pipeline, there are different labs trying to increase our understanding and advancement. And like I would say, as the farther you go down on the pipeline, the more complicated this gets. So yeah, most of the early stuff, like from 2000s to today, has been driven on the first step, the DNA step. And we're now starting to like move into these like more downstream steps of the dogma.
And I think this is where we bring Josh in just a little bit, and let's talk through like, okay, I am now staging up multiple kinds of data in
Yeah.
increasing size and complexity, and starting to try to look at how am I going to get a cross-functional view of all the different things that are going on.
Yeah.
Um, Talk to us just a little bit about the challenges when you have these quantities and different shapes of data, and what that's going to do to the machine learning approaches that we wish we were applying.
Sure. So I guess it's interesting because like, when people start to think of like machine learning and these AIs as like magic, right? Like they're just gonna you give it data and it's just gonna do it. Unfortunately, that's not the case. And they're really smart, but especially when we're talking massive data sets that may not even, but like also that may not really match each other, right? Like there's this, like a machine's not going to really understand how to interpolate that. into something meaningful. Like it's kind of like, if we're taking the recipe idea, you've got something coming in that's like a baked good, and you got something else that's coming in that's some, like, I don't know, like a savory chicken dish or something like that. If you tried to like mash those together and mix them up and try to create something from that, you might get lucky and you might end up with like an English pasty or something, but odds are you're probably not going to end up with something edible, or usable in the reality of it. So that's right. It's kind of interesting that you can't just magically like throw this data together and expect kind of a resolution. You still need kind of that really that human component, right? You need someone in there that can think, that can I'm trying to think of the word, like ideate, like that starts that spark of inspiration. That's the word I'm looking for. Um, and then once that person kind of figures it out, or like has an idea, like, it's like, okay, if I do X, then I do Y, then I do, or you follow the recipe, and it's like, cool, I can make this happen. Um, and that you make it repeatable. Well, now that's where a machine can really easily come in, because you can tell it, hey, if you do this, you give it the full recipe, like just a little box. You say, now go do this, but maybe your base ingredient changes. Maybe it's not chicken, maybe it's beef, maybe it's whatever. But like the rest of the recipes, like you can be reasonably assured that you're going to get something edible at the end of it. Very similar, we can apply it in kind of these cases where these like massive data sets, or I guess like genomic sequences. But there's like a pattern that we're looking for, or there's like a set instruction that we want to keep reapplying to all of these things. The AI, or sorry, the machine learning algorithm is going to be able to do that way faster and with a lot less like error, like a lot less error prone, than a standard human. So that's where it's kind of cool. I know, Grant, we've talked a little bit about this kind of in the past, so I don't want to gloss over that. Um, it's, but there's like I forget the number you said to me, but there's like a lot of possibilities, like genomic sequences out there that we've kind of mapped already. I don't remember, there was some big number we talked about in the past, but but like, so if we had to try to apply the same process to all of those to try to map, like, or just not even map, but see if the right pattern exists inside of it was the RNA or is the DNA at this stage? To kind of like
DNA, yeah.
DNA. Okay, thank you. And it's like, is this sequence in the DNA? Is there any potential here? If you had a human doing it, it'd be a lot slower. And they're tired one day, they might miss something. But that's where machines are really good. They don't get tired. They'll just keep flying through stuff. But you still
Sure.
have to have already discovered it, right?
Yeah, I can kind of build on that. So to kind of explain, right? If we think back to my history analogy, right? My grandparents' generation, they were finding all the drugs, they were finding the compounds, but they didn't know how they were made, right? They just knew if we extract this from this organism, we get this drug. The generation after them, right, they were the ones that were taking those known drugs and then doing all of the tedious work to figure out, okay, what are the genes actually responsible for making this compound? And that was like tedious work that's been going on for decades. But in that is kind of a bias, because you only are characterizing the types of pathways where we already know the compound. And so kind of getting at to where I said, hey, we think only like 10 to 20% of metabolic pathways are really turned on in a lab. Most of the ones that we've characterized are the ones that are active in lab settings, because they're the easiest to go after. If something's not ever turned on, you don't ever know what you're searching for, like when you're doing the experiments in a lab setting. And so there's like this inherent bias in the data sets. And just like, it doesn't matter what your application of AI or machine learning is, if you have bias in the training data, there's going to be bias in what the machine can predict. And so
that's really interesting, because it's almost like, by relying on the technology itself to kind of do the work for you, we're starting to like miss potential discoveries, or miss, like, not necessarily out of laziness, but like, it's trying to think, like,
you reinforce
wrong
the
assumptions.
things you reinforce the things you already know.
Right.
And I guess sort of two questions in that domain. One is, are we finding more things more potential classes or not potential classes, more candidates that might be biologically active and interesting within those existing classes? That's sort of question number one. And then question number two is, how do we start to break out of that and
Mhm.
not be looking in the same places every time?
Sure. And yeah, the first answer is yes, we are. And we also aren't at the same time. So most of the work that we do is in this DNA realm, where we have a genome and we're trying to find potential drug pathways. And the machine learning algorithms that exist, because we do have enough data that people have done AI and machine learning on the DNA side, they're good at finding, it's almost like different flavors of what is known, right? So there can be some novelness, but if
Mhm.
it's like way different, like different enzymes that we've never even known could be in a part of a pathway, they're going to be missed. And so it has been really helpful. And this is kind of what Josh was referring to. There are tons of papers today where people get thousands of genomes. They run what's called genome mining on the genome. So it's trying to find all of the drug pathways, and you get like millions of potential pathways where only like 1% of this space has ever been explored, right?
Yeah.
Um,
Actually,
this is
question on
where
that
we're,
real quick before
yeah,
you
go ahead.
go too far. Yeah, so because you talked before too about like we're kind of like rediscovering things, and it's maybe a duplicate and maybe created a different way with a different source mushroom or something, um, or fungus. Is that are we also discovering new things still with the
Yeah,
kind of
we are.
same patterns? Okay.
Yeah. So a lot of the when I'm referring to like things on a more like technical level, it's like chemical classes.
Okay.
And so different chemical classes will have different enzymes that they require to be made.
Sure.
And so they'll all have the same starting enzyme usually, because you need to start from some point, but then maybe they have other modifications that change it downstream. And
Mm.
so that we're really good at finding. Like if you have
Okay.
one starting chemical class, and you can have wildly different functions of different enzymes that have, sorry, two different compounds in the same chemical class can have wildly different functions. Um,
Interesting.
like for example, there's a chemical class called non-ribosomal peptide synthase, whatever jargon. It's a class, and one of the compounds in the class is the most toxic compound made by a mushroom, and another compound in the class is a drug that we use all the time in humans. So, like a chemical class alone is not going to define the biological interaction that it'll have on an organism.
Sure.
And actually, we have no way of going from like structure to predicting biological interaction at this stage.
Interesting. James, you're smiling. You're about to ask why.
No, 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 I'm not. Actually, I was gonna pause here and think just a little bit about, now that okay, we went down sort of the first pathway and said we are still finding interesting new chemical compounds by mining the parts of the genome that we know about, looking at the metabolic pathways that we know about. But you referred to sort of that void where we really tailed off in the number of
Mm-hmm.
drug discoveries that we've found. Yes, the genomics era helps, but we're still over on the left-hand side of your diagram. We're really still at the DNA step. And what you just said is, I don't know all the different metabolites that are going to come out, and I can't predict all the biological interactions.
Yes.
So the payoff is over on the right side. I need to find those compounds. I need to screen them. I need to see if they are biologically active and interesting as a potential drug.
Mhm.
Talk us through some of the, like, how do we start to move out of just the DNA phase of this and get into some of the downstream payoff of, yes, I can now find new drugs that are maybe part of a new class.
Sure. Yeah. So your summary is exactly how I would say it. Like we've gotten good at this DNA thing, but really we care about the more downstream steps. Like we want to know what the biological interaction of a metabolite is. Um, sorry, I lost my train of thought. Yeah. So basically, this is why I think a lot of companies are failing today. So, right, there was this bust period in drug discovery genome sequencing took off, people realized, oh my gosh, there's so many pathways. And a bunch of companies in the USA started getting seed funding of like, wow, we think we can find new natural products because look, there's all these pathways, we can see them in the genome. But really what a lot of companies are running into, and like different alums from my lab that work at these companies, that's not the real bottleneck. Like you can find millions of pathways, but if it's very difficult to screen and like figure out what the metabolites are, like basically that's the bottleneck. It's so hard to like in a throughput manner characterize compounds from crude extract. And maybe that's a little too like jargony. But the bottleneck basically is further down in this step, but we're much earlier on in our like advancement as a scientific field on these more downstream steps. And so we're kind of entering a bust era again where people are realizing DNA is not alone good enough. And I think it's going to be advancements in the protein and metabolite data sets and advancements in like getting AI going in those steps that is going to lead to like a second boom, a new revitalization of natural product drug discovery.
So,
But
I
that's
guess
my
sort
opinion. This is very
Okay.
like Grant's opinion. This is not like
We take
fact.
opinions around here.
we take lots of opinions. So I guess my understanding of the in the I'm not a crazy scientist version, I'm a technologist. Right now, well, I mean, I understand from like the modeling perspective, like we just don't have enough data for a model to predict something. So I guess a little bit on that, just real quick, is like, in order to train the machine to understand something, like we have to have enough, we have to be able to repeat something enough times in enough different ways for it to be able to predict an outcome,
Yeah.
based on, and that set can be very broad and varied as long as there's enough data to kind of support and train that. And then we're just like lacking that right now. So I guess
Yeah.
and it's like, and I guess where's that gap? I guess, is it like, we've identified the sequence. I guess, is it at the point where we're trying to trigger it in the lab to create something? Is it like we've created the something, what do you call it? The not the chemical, but the like you've produced you've triggered the sequence, you've turned it on, it's now produced something. And then now what that does in the human body is kind of where the big gap is, or is it farther down that line, or?
I mean, all of these are potential bottlenecks that academic labs are working on, but I would say the big one that companies specifically that are doing like throughput screens with robots, like more than just a grad student are running into, it's all it's the metabolite side. And I do have a hidden slide that I was going to maybe show because I think it's helpful to like demystify what I'm talking about and show you guys. It's because the data is more complicated. That's all it comes down to. And so Ray, maybe go to the final hidden slide that I had. I want you to, this is literally what I'm talking about when I say data. And so on the left, I'm showing what like me, a scientist, sees when I'm talking about genomes. It's just literal string text. And so all of the like tools, bioinformatic tools that have been developed are just basically glorified string parsers. It's not really actually that complicated. Like, yes, you need to know the
part of it.
biology, but like it's not that hard to, it's pretty simple, right? It's just strings. Metabolites, it is way more complicated. And so I'm just showing you like what the literal data looks like when you run the most cutting edge type of machine. It's called mass spectrometry. So on the bottom right, this would be like what you see for all of the metabolites in an organism. You get all of these like peaks, and these peaks represent different compounds that are like crashing out in your machine, but multiple compounds can be crashing out at the same time. And so that adds a layer of complexity. And on top of that, if you click on any individual point, so on the x-axis of the bottom right graph would be time, there is a spectra contained within that spectra. And so the top right is like an individual data point for one fraction of a second on the bottom right. So just imagine how complex this is. You have a spectra and then spectras contained within spectras, and just the nature of the data itself. Like we do not have tools that can go from what you're seeing to we know what metabolite is in there without like months of effort and labor from like some chemist. And basically the fact that there is no throughput way to do that is why there's no throughput way of doing drug discovery, because it still is slow.
And I guess at this point we haven't even touched on like what this thing is going to do in the human body, right? Like,
Oh God, yeah.
yeah, like that's like a whole nother like depending on single biological cell or some interaction of cells in your body, like what even happens.
Yeah.
Um, that's crazy. Yeah, that's cool.
I mean, that's even more complicated. So I
I was
think
gonna say like,
I,
that's it sounds like we're going like stepping scales of complexity.
it is,
Uh,
and each one builds on each other, right? So we're getting pretty good at the DNA thing. I think in 10 or so years, we're going to get better at the metabolite thing, where
sure.
hopefully some tools are developed where you can like take this complex data that I'm showing you and actually get predicted metabolites simply. But then
Right.
people will still struggle with knowing the activity. And I think that's not going to be for a long time. Because really, if you can predict activity from a metabolite alone, you could do computer generation of new drugs. I mean, that's like the holy grail of AI solving all of our problems. But it's
That's also oh
really,
sorry.
really, really hard. Yeah.
Yeah, and I say at that point you're also I mean you would have had to known enough about the human body itself and its diversity and its like to be able I mean essentially at that point you're saying like a computer understands us better than like we understand ourselves kind of
Yeah.
thing.
Essentially, like it knows what a compound's going to do in the body. Which, yeah, there's
Right.
so much complexity to that question.
Right.
And to be clear, one of the reasons that we're interested in things like AI in this space is it is better in high dimensions,
Yes.
right? One of the advantages of using AI to examine these problems is if you have rich enough data, but it's very high dimensional and very rich,
Yep.
it takes a significant amount of effort to mine it and distill it down to a place where the number of variables that you're dealing with fits nicely in a human head.
Yeah.
Mhm.
I can work in higher dimensions if I let the computer do the higher dimensional
Yeah.
configuration work, right?
Yeah.
But
Like I
this
was gonna
is
say with like the visual here, like the computer would understand this really, really well if we had enough data to like help it predict. Like it, a human reading this is going to take a long time for every single, run or every individual like point on that, sub-second timeline. Computers can do that and like snap of a finger, but it doesn't understand like a human does what this means at all. That's I guess that's the understanding piece is what we're missing, from the
Mhm.
computer at this point. Um,
and this is now you are into the scale in the laboratory
right.
problem. Where we start running into issues here. So Grant talked about it is hard to differentiate all the different compounds that you get out of a crude extract. So I have caused my organism to produce a bunch of compounds. I have screened them and gotten out this wildly variable set of mass spec. And now I'm trying to characterize all the different compounds that are in there and then screen through them and find the ones that are biologically active.
Yep.
Mhm.
If we had infinite dollars and could set up the massive scale experiments, yeah, wouldn't that be nice?
Yeah.
Then you could collect enough data where you can start to triangulate it even in these higher dimensions.
Mhm.
We don't have the infinite complexity, so you need to be mining a more targeted subset of these. And that I think is sort of the question I want to ask Grant. Why don't you spend just 2 minutes talking about sort of your research, why fungus, and some of
Mm.
the original stuff that you have done. Let's go down that rabbit hole just a little bit.
Good
Sure.
rabbit.
Yeah. Talk about my role. Can you go
Yeah.
back to the central dogma slide, Ray? That one right there. So yeah, my lab. So natural products is complicated enough that we usually labs will silo and specialize in a type of organism, and that's just because of practicality. It's like too difficult to be a plant natural product person and a fungal. So my lab is like probably the leading fungal natural products lab in the country. My PI made some very large discoveries on like toxins that are in our food products like years and years ago. But what I specifically work on is the DNA side of the world. So the side that I think is not the bottleneck. We're still getting better at it. Um, essentially my lab found a new type of natural product. And I mean like completely new, like it was a new chemical class. But it was so new that it had never, like the types of genes that make up that pathway had never been incorporated into the algorithms that we use to predict drug pathways in DNA. So it's just completely invisible to us, like as a natural product community. You could not find them in genomes. And so I made my own algorithm that was specific for finding this new class. And so, yeah, even though we are really good at finding metabolic pathways within genomes, there are still things that we are finding. Like there are still new discoveries. And in the case of the pathway that I found, once I made my algorithm and I ran it on every fungal genome that was in the world, we ended up finding that it's the 5th biggest class of drugs that fungi can make, relative to like 20 some classes. So number 5, and it was just completely blind to us because this class is just not normally turned on in conventional lab settings. So it just went under the radar for a long time.
All right, vanity question to follow it up. What's your favorite fungal species to work on?
Ooh,
My favorite fungal species to work on. I would say Trichoderma is my
Any
favorite.
particular reason?
Trichoderma is the fungus killer. So it is a fungus that infects other fungi. And it is really interesting to work with. It's a powerhouse for producing enzymes. And so there's a lot of companies in the USA that utilize it to basically produce like compounds, perfumes, different things that we use, like laundry detergents. Most laundry detergents that you buy are made by a company called IFF that synthesizes it from Trichoderma. So yeah, it's just really cool genus.
out of curiosity, is that the one where we get like the antifungal, antibacterials from? Like, is there's like in like medicine, like you've like a normal antibiotic, like it doesn't work on everything. You need like specific ones need like an antifungal version of it or something.
Um,
Whole
I'm
different
not
class of compounds.
fully understanding the question. Yeah.
Oh, I don't know. This is something my wife's an RN or an
Oh,
NP.
like, do you need specific? Oh, I see what you're saying.
Yeah,
Like,
yeah, yeah.
is
It's
it
like
one of the antifungal resistant strains? Is that what you're
yeah,
what?
something like, or like, there's like a certain type or class of like antibiotic you give to certain diseases that are like based on like fungal spores instead of, yeah. I don't fully
Yeah,
understand the topic because that's her domain, but
for maybe what it is that so with I don't know if this is answering your question, but I'll just go on a little
yeah,
rant here. We are way better with antibiotics and antifungals.
okay,
And that's because if you look at the tree of life,
sure.
fungi are actually more closely related to you and I than they are to bacteria, which is
Interesting.
counterintuitive. Most people would assume, oh, bacteria and fungi are both microbes. They're probably more similar, right? But no,
Yeah.
we share a lot of our like cellular structures with fungi. And so if you think
Interesting.
about what makes a good drug target, you want to attack something that is in that organism, but not in you, right? And
Oh, sure.
bacteria have a lot of unique structures that only bacteria have, but the human body doesn't have. And
Okay.
all of those make perfect targets for an antibiotic. But because
Mhm.
fungi are way more similar to humans, right?
Mhm.
There are way fewer things that you can go after that's not also going to be self-toxic. And one of the
Interesting.
only things basically that fungi have that humans don't is a cell wall. And so there's only like basically 2 major classes of antifungals. Both of them go after only the cell wall because we've never discovered any other antifungal. Everything else is going to be toxic to human cells as well. And it means that we're way worse at like dealing with fungal infections and bacterial infections. There's way
Huh,
less drugs to like use in a hospital setting.
that's interesting. So I guess with these, you talked about like there's still probably undiscovered pathways and things. Is like is part of that like if we like kind of go down there, I'd say there's like another category that's discovered. Like, would that help us maybe identify differences between the two, or would we still be looking like is that like another area of research we need to find a difference first, and then we can kind of find a way to attack it?
Yeah, I think that's the difference between like what compounds are being made and what they're doing, right? And so
Right.
every time that we find new classes of drugs, it's
Yeah.
increasing the pool of potential drug candidates. but
Sure.
that doesn't mean that they're going to be doing a certain thing, right?
Yeah.
But it's increasing the pool. So it could be that, yeah, the new antibiotic is within that new pool, but you still would need to test that compound on whatever bioactivity you're hoping to find, whether it's an antifungal or anti-cancer or whatever it is.
And then I guess one more, because I mean we're past technology being able to help at this point until we map the entire like human gene or human body. And but if you're like, you discover something that like the machines are missing at this point because they don't know the pattern to look for it. Like they just that human knowledge wasn't around when they were trained. So I guess when you guys are doing this, like, is this something where, in the lab setting, you were talking about like they just don't turn on in a certain way just because they're comfy on their couch, watching Netflix and eating nachos. Um, is there a way to like, I guess, how do you change that? Is there like you're going to
Oh,
rainforest and just being like plucking it up and trying it out, or um
yeah, there's whole, I could go deep on that. I mean, people have been trying to
fair.
solve that for a long time. I mean, the biggest thing, there's a whole strategy called OSMAC, where it's called one strain, many compounds. And
Okay.
so you take this is like the main approach people use. You take your strain and you grow it on as many different media conditions as you can, because one
Hmm.
media condition, for whatever reason, might turn on a pathway that's turned off in another media condition. Or maybe you take your strain and you put it in competition with something. Like maybe it needs to be fighting against a bacteria for like the antibiotic to turn on. And people have also used that with success to find certain things. The more like advanced approach is you literally genetically engineer your organism to like overexpress the pathway that's not being turned on. And that's the most laborious, but it also is like what a lot of academic labs are doing to kind of push the field forward today.
That's interesting. Yeah, and I guess just from the tech end, I think it's fascinating because like, we can't teach a computer what we as humans have not already learned, right? So it's like I think it's a novel idea to like think, okay, well, what if we just like kind of mimic this in a virtual space and then ask it a question? It's like, well, until we actually have the data to of at least something similar enough, or and enough of that to reasonably be assured on like what the outcome can be, like that the model or the computer has no idea. Like it's not like you have a virtual space that's been created out of nothing and you can walk
Mhm.
around in it and like throw something in a machine and just have machine tell you, or like have it go, yes, this is what it's going to turn into. This is what it's going to turn into being. You still have to like do the physical and watch like how nature actually happens. And I think that, yeah, that's fascinating to me. It's like, but then once we do it a couple million times, or whatever the data points need to be, that's when we can kind of take that next step into, okay, maybe we build a model that's 60% reliable, and then we learn
Mhm.
more and we run that model, and okay, we figure out why it's failing, and now okay, now it's 70% reliable, and like um yeah, the future is going to be
Sure.
crazy, but it's kind of built on the backbone of sweat and labor. So
Yeah,
That's
I mean that's kind of my like summary pitch, I guess, of this whole
yeah.
thing. It's like machine learning and AI is very useful, but we always, no matter how you're applying it, you have to be aware of the limitations of the training data. And in the case of drug discovery, we're
Mhm.
only really able to apply it at the DNA step at this point. And it's my personal opinion that it's impossible to apply it for the protein, metabolite, and downstream steps until there's just more fundamental scientific advancement. And
Yeah.
this is a pitch to fund science. Please keep funding science. Like
QR
basic
code on the
science is
screen. No.
so important. And it's, I mean, actively getting kind of cut right now. So
Yeah.
like US industry is not going to be able to like make new drug discovery companies unless we like just fundamentally understand proteins and metabolites better. And the AI models are not going to solve that. We need to just fundamentally do the work as humans first.
Mhm.
I love that you guys went there naturally. I was gonna try and ask some questions to get us there. We are closing in on sort of the end of our time here, but while we're waiting to see if we've got any questions coming in on the from the audience, I guess I would ask sort of the follow-up one there. We talked a little bit of the far future and like what it looks like if we have those masses of data. Um, let's start just a little nearer term. Grant, you talked about where you're seeing some of your colleagues and folks that graduated from your lab that are now out in industry and where they're finding the bottlenecks. You've talked about maybe we're nearing the end of what we're going to be able to find with the DNA level of this and
Hmm.
need to start shifting to the right in your
Hmm.
diagram here. What are our short-term opportunities? What's the thing that moves us forward over the next year or two?
I really strongly believe it's going to be like public universities. Um, I think the investment community has soured a little bit on natural products because there was like three big companies, Zimmergen, LifeMind. I mean, I have alum that are all these labs. Hundreds of millions of dollars was poured into these companies, and they all said we're going to find the next drug, like using, and none of them have. That's the summary. For whatever reason, it's more complicated than all these people thought it was going to be. Just having the DNA like component has not been actually helpful for finding the drug candidates. And I think the investment community sees that and is kind of going to sour on putting more money into natural product companies until there's like some large tech advancement. So academics are going to keep doing what academics are doing. We're going to keep doing this work. We're going to keep doing research. And I think that is where there's going to be some big advancement from academics that's going to enable industry to try again. And that's my, yeah, that's my opinion.
No,
So I guess
and again, we take opinions around here. Let's
once
ask
that happens, though, actually curious. So, like, the academics like make the initial discovery, and then your industry takes over. At that point, is that when you imagine like, it starts to like just kind of take off and like, or what? Like they leverage that discovery and just kind of launch it, or is it kind of still?
Hopefully, the academics, we don't have the scale to do things at a like it's the throughput, right? So we can like
Right, right.
proof of concept something on like you have one grad student doing it by hand. It's industry who can take that proof of concept and run it through a robot that's doing it like 1,000x, and
Sure.
like just the scale of the screening is so much bigger. And I think that's, but we have to show that we can like, it's basically going to be, can we actually predict metabolites from crude extract? That's going to be the
Yeah.
big thing. And if you can
Gotta
do
prove a
that, there's
concept
going
at
to be a
first, and
company. Yeah.
So
interesting.
you just set the impossible challenge, which is we're going to go back to our academic silos and continue to work on the fundamental science or science, and we need the scale and the throughput and the investment dollars to drive through and generate the huge data sets.
Yeah.
Th-
We got
There
a
is
very technical question in the chat. Someone's a natural product scientist.
go for it.
So I don't know if this was sent to everyone. Stephen asked, what do you think the current potential for predicting non-ribosomal peptides with AI is? I can tell he does natural product research. So non-ribosomal peptides are a specific class of drugs. And Stephen, I think you're talking about like the A domains, really. And I'm going to go so deep in jargon, I don't know how to explain this to other people. But yeah, A domains dictate which amino acids grabbed from the pool of primary metabolites. And we actually are getting okay at that. There's a new tool that was developed by the Marnix Medina lab, where it attempts at predicting amino acid specificity from those A domains. And it's pretty good. I think actually we're going to get better at NRPS and PKS predictions than the other classes first. That was a very technical question. Thank you.
No, I love it. We've got geeks in here. Welcome. You have found your place.
Yeah, well, one of us.
But
Um,
yeah, yeah,
no,
sorry,
go.
go ahead.
No, please.
I was just going to say, if that's research you do, Stephen, look into the Marnix Medina lab. There's a new paper. I think they're trying to print it, like publish it now, but it's going to be like a program that you give it an A domain and it tells you which amino acid it's going to grab.
Interesting.
I think I'm going to wrap up by asking Josh and Grant, how do you reconcile that impossible problem that Grant has just set us? How do we do the fundamental research and do it at scale to get the data that's going to drive this next wave of discovery we all think is there, but haven't seen yet?
It's an interesting one because I think part of it has to do with like capitalism, or like flat out. But like especially in like the tech industry, a lot of the time we'll do proof of concept on something where it's not necessarily built for scale; it's built to prove a point. And like But once you prove a point and it generates interest, now all of a sudden someone's like, well, I can use that. Like that'll help me be more effective at whatever I'm doing. That's a novel approach to something that's something I can monetize, things like that. And then once you hit there, then all of a sudden you're starting to get the a little bit of that seed money to kind of start to scale and build something. And in the tech space, what we do is we modularize it so we can start to easily repeat it and like launch it at scale. From there, I feel like that's something very similar with a little bit more like physical headway you have to kind of build into, like the robots that are going to do this. But it feels similar where you kind of have to first have an idea that's going to generate enough capital interest in order to, kind of launch. And then, once it's gone from a handful of labs and university out to, a big pharma company or something along those lines, all of a sudden the data starts flowing in and stuff. And I guess in the reality of our world, we still have a little bit of a sharing issue. We don't play nice. Like, there's some piece like, Epic has this huge Cosmo database that they do share with researchers and stuff like that. But it's not like, private industry. Like I can't go to Epic and be like, hey, give me access to your database, So there's a little bit of that sharing issue still, but at least in the I would say in the broader strokes of like human interest, it's available. But there's because everyone wants to monetize and make a profit on some of this stuff. So I think that's kind of the hitch, is like finding something in the generalized space that someone can make enough money off of, which will get us then the data to help kind of back in the beginning stages and start to then discover more, and then which some of it may be valuable in that kind of capitalistic sense, but then some of it may not be, but would be good for, super niche life-saving treatments, stuff like that. Like I said, that again, opinions. We like opinions here. So um I don't know if you have a different thought process on that one, Grant. There,
No, it's well, I don't. What was the question again, James? I don't remember.
Yeah.
So I was asking for sort of the wrap up thought. Like, you pose the impossible challenge. We need to go back to our little more siloed laboratories and nail some
Uh,
more fundamental science. And we need the scale. We need way more data than we've got. We need the high throughput experiments. It's going to cost a lot of money. Um, how do we reconcile those?
I mean, I would say either governments need to step up and keep funding science, or industry needs to realize it's not beneficial to them to hang on to everything. And in fact, I caused a little drama at a conference in January on this exact subject. So with fungal genomes, there are about 6,000 sequenced publicly available genomes. That's what I'm working on. That's the amount that I have to use, right?
Sure.
There's a company called Hex Bio in the area. This is just as one example. And they're a natural product drug discovery company that's trying to find things. They've sequenced 100,000 plus fungal genomes.
Wow.
Okay, the public domain has six.
Yeah.
This private company has 100,000 plus. And their pitch is that yes, with enough data, we can solve all the things that they think we can solve. I think they're going to run into the same bottlenecks no matter what. It doesn't matter if you have more DNA, but my fear is that HexBio is going to go under, which is being talked about, and that data is just going to disappear. Like, it's not going to be released to the public because investors are not going to want to make it available to the public. But yeah, either governments need to fund science or industry needs to realize actually by making things available to the public, it can benefit us in turn, because they will make advancements that enable future companies that we can't even imagine today.
That's
I
interesting. It's like
love
we all
that.
need to go back to preschool, learn to share again.
Sharing, yeah.
I
So
love
at
that.
the conference I was at, there was this company was giving a talk, and I asked the question. I said, "What are you guys going to do with your data if your company folds?" And everyone at the conference started laughing. It was a thing. He wouldn't agree to say that they would release it.
Probably did not have permission to say that.
No,
Yeah.
he did not have permission. Yeah.
I think that's where we leave it, is with a little bit of controversy and maybe some hope. Um, Grant, thank you so much for your time today. I'm going to give it back to Ray, and she's going to take us out. To the audience, thank you so much for your time in joining us. We've appreciated having you here. And everybody take it easy. Ray, take us out.
Awesome. Such a cool presentation today, you guys. Folks in the audience, thank you for joining us. Before you head out, we want to invite you to next month's webinar, Beyond Digital Twins, how software simulation is transforming lab automation and product development. A little bit about this webinar. As lab automation evolves, the need for simulation-driven testing and configurability is critical. This webinar will explore how digital twins and software simulation optimize product development, enhance automation reliability, and create scalable test environments. If this is something that interests you, go ahead, scan that QR code on the screen. It'll pull up the Zoom link to register. We'll also be sending out a follow-up email with a recording of today's presentation and information about this upcoming presentation. Thank you, Grant, Josh, and James. Everybody have a great day, and we'll see you next time.